Today we have one of the most exciting, timely topics that one can think of in medicine or biology, and that's sort of where genomics interfaces with clinical disease. And we are very fortunate to have two investigators from here at NIH whose work has been at the forefront of this. Somebody asked me the other day, why do you keep showing a picture of the Brooklyn Bridge? And I guess I could put a picture of my wife or grandchildren there, but it's one of the objects that I find myself in love with, and symbolically it represents the purpose of this course. If you're ever in New York and want to do one of the most exciting things, and it won't cost you any money, is to take somebody you want to be with and walk across the Brooklyn Bridge. There's a wonderful bar under it on the New York side and a wonderful restaurant on the Brooklyn side, and the view is unbelievable, and the history is fantastic. But it's to connect two worlds, and that's the purpose of all this. So if you could, Jeff, could you flip on the first slide? So what's so exciting about today? From my way of thinking, what sort of provoked this was reading a bunch of editorials and commentaries by learned people about the question of, is there a new cancer paradigm? The old paradigm being based upon the fact that diagnosis, to a great extent, was based on morphologic criteria. So if a cell looks malignant and it's from a colon, for example, it's a cancer of the colon. And it was based on these criteria which were graded by severity. But now it was observed repeatedly that the clinical course, the response to therapy, really didn't correlate all that well, except maybe in a big statistical sense among groups of people, but on an individual basis, there was a lot that was going on there that wasn't revealed by conventional or even specialized immunocytochemistry. And then when one enters the genomic era, this issue arises of whether these tumors, which are expressing different genomic patterns, not uniform, it may be a colon cancer in one patient expressing one pattern of gene expression, but in another there are other genes, other mutations, a whole new level of complexity that's arisen. And out of it has come the question of whether genomic and other analysis of tumors actually provides a more accurate diagnosis and permits individualized treatment and some prediction as to the clinical phenotype. What is this tumor going to behave like? And that's an exciting frontier. There are no answers at the moment, although our speakers are best suited to comment upon this. Okay? So the subject, if you could show the next one, Jeff, is a very extraordinary disease that you probably know a fair amount about, melanoma, or malignant melanoma, but it has some clinical mysteries connected with it. So here's a case report of a person very well known to me, happens to be a cousin of mine who died a couple of years ago. He was in extraordinary good health. He's an athlete, played tennis up until the time he was about 91, 2 or 3 times a week. And he was in great shape and took very good care of himself in all ways, nutritionally, all sorts of good things, no bad habits. And at about age 74, it says myeloma, it should be melanoma. He had a spot on his right shoulder. And it wasn't terribly large. It was maybe 1 centimeter, something like that. And it was biopsied, and it was a malignant mole, a malignant melanoma. And the surgeon did what should be done, was to remove a large margin around this. And then subsequent analysis, studies done over an annual basis by very competent centers, this was at the University of Cincinnati, had annual follow-up, including scans of the abdomen, various blood tests, all sorts of things, and they were all negative, and this was done routinely. At age 92, he observed that he was losing his memory. And everybody said, "You look, Bob, you're 92 years old. You can't keep doing this forever." And that went on for a little while, but he kept getting a bit confused. And that wasn't quite like him. And eventually, he was worked up in general, and nobody thought that this had anything to do with a melanoma. He had a convulsive seizure, which led to a neurological workup, and it was found that he had 3 roughly 1 centimeter tumor masses in his brain, mainly in the frontal and parietal lobe. This was biopsied, and it was malignant melanoma, and they had tissue from the original skin biopsy taken when he was 74 years old. And the pathologists and all at the University of Cincinnati went over it and said, "Looks the same to us." To the best of my knowledge, there were no genomic studies done on either of the tumors. Well, his course from then on was disastrous. He had no response to radiation or to medical therapy. He was kept comfortable, hospice, coma, and he died. There were no lesions found clinically in his bones or his liver or any other tissues. Now, this is one of the great mysteries of this disease, is that people can have a diagnosis made and the subsequent course is highly varied. This is the most extreme I've ever heard, but maybe you will comment about this. But it seems that 18 years is a long time. But nevertheless, one can't escape the fact that this tumor was doing something for 18 years. And it's highly unlikely that a primary melanoma was going to develop in the central nervous system, except for the eye. Can we make the next slide? So the last slide that I have are just some of the questions that pop into my mind when I realize what the topic is, and maybe they're in your mind, and they're intended to encourage you to come up with more and ask our guests. How actually does sun or tanning salons, how do they cause melanoma? And do they actually promote the development of malignancy? How does it happen? Is this a sunbelt disease? I mean, we all know that the epidemiology shows that in places where people sit around in the sun and all, that they're more likely to have it, but it's not exclusive. So there's some question about it. Now, recently I was in Australia, which is the #1 place in the world for melanoma, and there's a huge uproar in the public press and in one of the medical journals about sunscreens. And do sunscreens actually prevent melanoma? Do they prevent malignant melanoma? And what accounts for the highly variable clinical course, particularly this idea of metastases? And that brings us to where we are here today. Are there diagnostic, genomic, or other signatures which predict a clinical course, highly variable in patients, not always predictable, and the presence of metastases? So this is one side of the bridge. The other side of the bridge, of course, is where do the basic science advances influence us in terms of new ideas about the biology of the process and about development of therapies, prevention, or so forth. So, we have our two speakers today. The first speaker will be Paul Robbins, who graduated from MIT and has a Ph.D. from Washington University in St. Louis. After a postdoctoral fellowship, he came to the NIH, where he has been in the surgical branch of the National Cancer Institute. This branch is known as being at the forefront of the development of techniques based upon cytotoxic T cells to attack malignant diseases. And the case in point today is malignant melanoma, about which Paul will comment. Now, he has been collaborating in the past year or two, I guess, or more, with our second speaker, who is Yardena Samuels, who was in the Genome Institute, I guess still is in the Genome Institute, but officially she is now an associate professor at the Weizmann Institute in Rehoboth, Israel. So Yardena's training was in Cambridge, and then where? Yes, at the Hebrew University, and then she received her Ph.D. from Imperial College in London, following which she had a highly productive and very exciting postdoctoral fellowship with the Vogelstein group at Johns Hopkins. And she has been at NIH as a tenure-track investigator, and as I mentioned to you, after 6 years in that position, will be leaving us to return to Israel. But we trust we will all see and hear more from her during the coming years. So it's a great pleasure to have you both with us, and I'll be quiet. And so maybe if you would want to begin, Paul.
I want to thank you for inviting me to talk today. And I encourage people to ask questions. Please feel free to ask questions during the seminar. I think it's better to get the questions out when they occur to you. So the title of our talk is Genomic Paradigm for Cancer Diagnosis and Treatment, Melanoma. An overview of my talk here is first I'm going to talk about melanoma prevalence and etiology, a little bit of background on the disease, and then review some of the current treatment strategies, and then finally, talk about developing novel treatment strategies that are based upon targeting non-synonymous mutations that change the coding region of proteins present in tumors. So, melanoma is a fairly prevalent cancer. This list here shows you the number of new cases of a variety of tumor types in males and females. Melanoma of the skin here is number 5 for males, number 6 for females. There are about 76,000 new cases per year predicted for 2012. And this actually has been increasing. So the numbers are going up, and actually they've been going up steadily for a number of years, as you'll see in the next slide. And over 9,000 deaths per year, and that number is also increasing despite a lot of work by us as well as others to develop effective strategies for treating this disease. So, as I said, the melanoma rate has been rising. This graph here shows the trend over more than 30 years for melanoma as well as a variety of other cancers. There is a lot of variability in some cancers such as prostate, but that may reflect the frequency of screening for prostate cancer, which of course is often done by, screening PSA levels. But you'll notice that some diseases, the frequency of some diseases are actually going down. Lung cancers, colorectal cancer actually is going down. Colorectal cancers are trending down, presumably because of, again, better screening. Lung cancers have been going down actually for a number of years in men, and they're leveling off for women, which is probably a reflection of the fact that men had been smoking cigarettes for longer than women, and so the women are just catching up, but presumably or hopefully this curve will be trending down very soon for women as well. But melanoma actually has shown a fairly steady increase over the past 30 years, and in men as well as in women. So it's a serious disease. Malignant melanoma, metastatic melanoma is a very serious diagnosis. So we need to come up with better ways to treat it, but we also need to come up with ways to prevent people from developing this disease. So there's a number of ways to accomplish this. One is go to see your dermatologist. Certainly stay out of the sun, certainly avoid tanning booths. It's very clear that, these things contribute to melanoma, and that's evident at the genomic level. As Yardena will discuss in more detail, the actual patterns of mutations have a signature that is known to be caused by ultraviolet radiation. So there's no question that the UV rays contribute to melanoma. Now, of course, it's a complex disease like most diseases, so environmental factors as well as genetic factors influence the development of melanoma. But nevertheless, that's certainly a contributor. Now, there's a number of factors that dermatologists take into account when trying to decide whether or not a mole, which is shown here on the left side, is truly a mole or is a malignant melanoma. One is asymmetry. You'll notice that this is a fairly round shape here. The shape is very irregular. The border, this mole has a very clear border, whereas this one does not. The color is of a mole is generally uniform, whereas a malignant tumor is not. And again, notice that just simply color alone, a darker color, is not necessarily diagnostic for melanoma. In fact, some people have very dark moles as well. So a number of criteria really have to be used to distinguish melanoma from a benign mole. Certainly the diameter is diagnostic, but one of the most important characteristics that's used in diagnosing melanoma is evolution. In other words, as these spots change in appearance or size, if that happens fairly rapidly, then that's a sign that could be melanoma. Now, it's not actually all that straightforward. There certainly are what are called benign nevi. There are growth of melanocytes, which are the cells that produce the pigment in the skin and give rise to melanoma. They can certainly grow. So the ultimate diagnosis really has to be made by a pathologist. But at least these are some signs that can be used to distinguish a melanoma, a malignant melanoma, from a benign mole. But the bottom line is go see your dermatologist. That I think is the key thing. Some patients actually have large numbers of these fairly dark moles, and basically what's done is patients' bodies are photographed. And they'll take a photograph, and then you'll come back several months later and take another photograph to track the progress of these moles. And if any of the moles are increasing rapidly in size or changing in size and shape, then that's used as a diagnostic tool for for melanoma. There's also a number of melanoma risk factors, of course. Certainly prior melanoma is one. If you had a malignant melanoma removed, then you're at an increasing chance of developing melanoma. If you have atypical moles, certainly you are at a greater risk, and you have sun exposure, extreme sun exposure, as well as fair skin with an inability to tan. That's also associated with melanoma. Your ability to tan is actually affected by your genes as well, because the genes that are involved in pigment formation, some of these have mutations that affect your ability to actually melanize. And so that can certainly be influenced by normal genes that are expressed in melanocytes and that are associated with the function of the melanocyte, with the ability of that melanocyte to produce melanin. But there certainly is a strong association with germline mutations. Now, up here I say that they're estimated to play a role in about 1% to 2% of all melanomas, but of course that number could be significantly higher as we discover more and more mutations that are associated with melanoma, and Yardena will be talking about this some as well. That number may, of course, go up. But the genes that have been strongly associated with melanoma include a gene called CDKN2A. A mutation in this gene predisposes individuals to develop melanoma at a very early age. So people that have this mutation, these mutations in this gene, can develop malignant lesions in their 20s or 30s, as opposed to what happens in general with sporadic melanomas, which generally develop much later in life. Patients typically that we see in our clinic are in their late 40s or 50s. And individuals that have mutations in this gene have a very, very high likelihood, a lifetime risk of about 80% of developing melanoma. So certainly family history is important, and that's why genetic testing is so critical for diseases like this, because if you know that this is a mutation that's present in affected family members, then you know your likelihood of developing melanoma is much, much higher. Now there's other genes that are associated with melanoma. CDK4 is also is another gene, which is a kinase gene, a gene that phosphorylates proteins, as well as mutations in a number of genes that are involved with pigmentation. Two of these are MC1R as well as tyrosinase. Tyrosinase is the gene that actually plays a critical role in synthesizing melanin. If you have a mutation in the tyrosinase gene, that can lead to albinism. So people that have no pigment at all in their skin frequently have mutations in the tyrosinase gene. But also other genes involved with pigmentation have been associated with an increased risk of melanoma. Not as high as mutations in these two genes, but nevertheless there is an association. But I think this slide really sums up the importance of screening and getting screened and catching the disease at a very early stage, because melanoma can be staged into four broad categories. Stage 1, where the melanoma is very thin, less than 1 millimeter in thickness, and hasn't spread to any sites. And patients with primary melanomas that are diagnosed at this stage do very well. You can see this is a 15-year time scale here. So a few of those patients will go on to develop malignant melanoma, but most of them will remain disease-free. If the tumor is, if the lesion is thicker, up to 2 millimeters, but still hasn't spread obviously to lymph nodes or distant sites, there is an increased risk of developing melanoma and actually dying from the disease. And then if it's spread to nearby lymph nodes but not distant sites, increasing number of patients will die from this disease. And the final stage, where it's spread to more distant sites, is uniformly lethal. Basically everybody who has developed metastatic melanoma to any distant sites will die of this disease. So what can we do about that? All right. So first, I want to discuss some of the current strategies that we have that are available for treating this disease. Now, there are a number of these. These are listed as strategies for treating patients that have stage 4 disease or unresectable stage 3 disease, meaning they have cancer, they have melanoma that it's spread to lymph nodes, but for one reason or another it can't be removed, it can't be resected. So if patients have stage 4 disease, they can receive surgery. In some cases, this is beneficial. If it's only spread to one site, it's possible that this may cure the patient, although in most cases, if it's spread to a distant site, there are micrometastases that may not be evident on X-ray scans to other sites. But it may, in some cases, benefit patients. Now, high-dose IL-2, or T-cell growth factor, actually was approved a number of years ago by the FDA, and this leads to complete responses in about 5 to 10% of patients. So merely administering a T-cell growth factor can lead to a complete remission of tumor. And I'll talk more about the use of T-cells in the following sections of my talk, the use of T-cells for treatment. An antibody called ipilimumab, which is directed against an inhibitory molecule, CTLA-4, also expressed on T-cells, can also be effective, although there are very few complete responders to this. Vemurafenib inhibits a kinase called BRAF, and this was recently approved. This causes dramatic regression of metastatic melanoma, but essentially no complete cures. So patients have remissions, they develop, nearly complete regression of all their, can develop nearly complete regression of all their melanoma, but then subsequently the melanomas recur. So by inhibiting this kinase, which is crucial for the growth of that melanoma will at least temporarily regress. But additional mutations then can occur in the tumors, and then they can, and then they recur in essentially all patients who receive this. Now, there's a lot of work on combining inhibition of this kinase with other molecules, other inhibitors of this signaling pathway, and there are some encouraging results. But nevertheless, we don't have many complete cures. We don't see many complete cures in patients that just receive this inhibitor. We're actually exploring the use of this inhibitor along with T-cell therapies in the surgery branch, and don't have anything to report yet. But that's another possible approach that may lead to better therapies. Chemotherapy really is basically ineffective. It was used for a number of years, but we know that has essentially no effect. Combining chemotherapy drugs with interferon or IL-2 seems to have no benefit. So basically, I think the hopes for curing metastatic melanoma certainly reside in the current and ongoing clinical trials. Now, there's a number of these trials. Some of them involve vaccination of patients, either with peptides, short amino acid sequences derived from antigens that are expressed in the tumors, whole cell immunization, as well as antibodies against another inhibitory ligand, PD-1, or PD-1 ligand. These antibodies interrupt the signaling pathway that actually inhibits responses of T cells to antigens, or adoptive cell therapy. And that's what I'm going to primarily focus on, because that's what we do in the surgery branch. This slide here basically just gives you a summary of the response rate to these reagents. Complete responses are defined as the disappearance of all radiologic disease for at least a period of 2 months. Partial response is a shrinkage of at least 30%, and this is the objective response rate, which is the sum of the complete and partial response rate. So you see in response to chemotherapy, you have very few complete responders. There's a few complete responders in the IL-2 group here, very few responders to anti-CTLA-4. There actually is a fairly high response rate, as I said, though, to the BRAF inhibitor. So nearly 80% of patients can respond to this inhibitor, but those regressions are temporary. Generally between 6 months and a year after the patient receives therapy, those tumors recur. And so we need better treatments. We need other approaches. Cancer vaccines are relatively ineffective, so immunization with proteins or whole cells really lead to no complete response. But T-cell transfer, the transfer of autologous T-cells from that recognize these tumors, actually does have a significantly higher complete response rate. So these responses, in contrast to the responses with the BRAF inhibitors, are very durable, can be very durable. So what I'm really going to talk about is the adaptive immune response to cancer. So this is the adaptive immune system, which are the T cells and B cells that can recognize foreign entities as well as molecules that are expressed on tumor cells. And this is based on an observation that was made a number of years ago, initially by Steven Rosenberg, but it's subsequently been analyzed by a large number of groups, that tumor-specific T lymphocytes are present within growing malignancies, but only rarely trigger spontaneous regressions. And that's the very rare event. Perhaps 1 in 1,000 patients do we see any kind of evidence for spontaneous regression of tumors like melanomas. These tumor-infiltrating lymphocytes, or as I'll refer to them as TIL, can be efficiently expanded to very large numbers, so 10 billion, 100 billion cells, and that's what we generally do in the treatment that we carry out in the surgery branch. And significant tumor regression has been observed in about 50% of melanoma patients who receive adoptively transferred autologous cells grown from that patient's tumor. Now, a second approach Okay, thank you. Now, a second approach that we're actively exploring, as well as a number of other groups are exploring, is to isolate T-cell receptors that are directed against tumor antigens and use these to genetically engineer T-cells to confer the specificity for tumor antigens on T-cells from that patient. And this will allow us to broaden the treatment to a wider patient population, as TIL can only be isolated from about 50 to 75% of melanoma patients and can rarely be isolated from patients with other cancers. So on this summary slide, I'll show you the approach that we've taken, which is to isolate tumor cells from a patient, culture those cells in vitro, and this is generally carried out in the presence of interleukin-2, which causes the outgrowth of lymphocytes from a patient. And then those cells, after in vitro expansion, are infused back into the patient along with IL-2. Now, the alternative approach is to remove peripheral blood lymphocytes from a patient and genetically engineer those cells to express a T-cell receptor that will now allow that T-cell to target the tumor cells and then transfer that back into the patient. Now, one of the things that we've found that seem to be very helpful is that we precondition patients. We give them chemotherapy either in the presence or absence of total body irradiation. And what we have found is that that basically provides space for these transferred cells to proliferate, to persist, to a much greater extent than in a patient who hasn't been ablated. Now this treatment, which consists of treatment with Cytoxan and fludarabine, which by themselves have no effect on the tumors or tumor regression, either with or without total body irradiation, basically that creates a lymphopenic environment where the number of lymphocytes in the peripheral blood drops from around 1,000 to 1,500, which is the number present in a normal individual, close to zero. And then the cells that have been expanded in vitro are infused back into the patient. IL-2 is generally also administered at this point to help support the lymphocytes that are infused. And you can see here actually that the lymphocytes don't appear to recover immediately, at least not in the peripheral blood, but they nevertheless do appear to undergo significant expansion. And we can actually, in some cases, see lymphocytosis. We can see higher lymphocyte counts than normal in these patients after they've received the transferred cells. And at this point in time, a relatively high proportion of the cells in the patient's peripheral blood actually consist of the cells that we've administered. Because we've ablated this patient, we haven't totally removed the lymphocytes from the patient. The patient still have some endogenous lymphocytes that do eventually recover. But a fairly high proportion of the cells in the peripheral blood in many patients can consist of the transferred cells. And we think this expansion provides an advantage in that we have a lot higher number of cells to cause tumor regression. Now, this slide summarizes our recent 3 trials that have been done using cell transfer therapy in melanoma, where we've transferred TIL from the patient either in the absence of total body irradiation or in the presence of 200 centigrade or 1200 centigrade total body irradiation. And you can see these numbers here are the number of patients responding, and then listed below are the duration of those responses in months. So a number of patients have partial responses. In other words, some of their tumors regress. They regress to a significant extent, but then they later on recur. So if there's a plus sign here, that indicates that the response is still ongoing. So these patients have all eventually recurred. Some of them actually have had very long-lived responses before the tumors have started to grow. So this patient here went for 7 years before the tumors started to grow. But a number of patients in this trial have had now complete responses. And you notice these responses are ranging generally between 5 and 9 years. So these are very long-term responses, and these are complete responses. There's no tumor detectable in these patients at this time. So these treatments can be very effective. As you can see here, 40% of the patients that were treated in this arm of the protocol have had complete responses, and all but one of them are ongoing for 5 to 7 years. So these are very long-term responses that we see in these patients. Now, just to give you an example of some of the characteristics of the melanomas as well as the response, here's a large lesion on the shoulder of this patient here prior to therapy. 12 days after treatment, that tumor is gone. That tumor has now regressed probably completely. Here, there's no real evidence of tumor there. We can also see complete regression of extensive visceral lesions. So this is a patient who had multiple metastatic lesions in the liver, and you can see it's just studded with lesions. And this is just one slice of the liver. If you look throughout the entire liver, there would be many more of these metastases. A month after the treatment, the patient's liver appears to be free of disease, and this is now more than 2 years after treatment, and this patient is still disease-free now 7 years after treatment. So the treatments are effective, they're long-lasting, they can be effective for visceral as well as a subcutaneous disease, and even very large lesions, such as this lesion on the scalp of this patient. This patient actually received a treatment in 2005, and you can see here now the patient is disease-free. It actually regressed much more quickly than that, but I'm just showing you this slide here to show you the durability of the response. The patient is still disease-free. There's no cancer in this patient. So relatively small numbers of cells can treat large, very large lesions on these patients. So what else can we do? How else can we treat these cancers? Well, another approach that we've taken is based on isolating the T-cell receptors from the reactive cells of a patient and then transferring those to another patient's T-cells. So T-cells recognize their targets primarily through the T-cell receptor, and this confers a specificity of recognition of the antigen, which here is presented as balls in this MHC molecule. And it's basically like a lock and key. This receptor has a great deal of specificity for the specific peptide, which is usually between 9 and 10 amino acids, that's bound to the MHC molecules of that individual. And so that allows us to create a cell that has a high degree of specificity for the cancer. It will not recognize any other cells. It will only recognize that particular molecule. Now, this approach initially was carried out by targeting melanocyte differentiation antigens. Now, these are genes that are highly expressed in normal melanocytes. They're expressed in normal cells in our skin. And many of these are actually the molecules that are involved with forming pigments. So there's a number of genes, many of these are enzymes, that play a role in synthesizing melanin, and variations in these genes affect skin color and hair color. So mutations in a certain enzyme in the pathway will lead to red hair or fair skin. So they play a very important role in determining your susceptibility to cancer, but they also, these very molecules can serve as targets for the immune system. And we initially focused on these melanocyte differentiation antigens as we knew that many TIL recognized epitopes derived from these proteins. This is actually a sort of graphic illustration of how these melanocytes can be targeted. This is a patient who received therapy with TIL, and this is his face shown under a Woods lamp, which is basically black light. It's a UV lamp, and the areas of the skin that are bright, that appear to fluoresce, are areas of depigmentation, where the melanin has been lost from the skin. And so you can see it's extensive. It's not complete, and this is not something we understand. We don't really understand why there can be such a apparently random pattern to it. But nevertheless, this can be one of the consequences of treatment with our T cells. And in fact, there's an association between depigmentation, between vitiligo and the therapy, and the response to therapy. So we initially targeted some of the melanocyte differentiation antigens that we knew were predominantly recognized by TIL by identifying a tumor-reactive T cell, these tumor-reactive T cell receptors, cloning them into viral expression vectors, producing retroviral supernatants, and then using that retroviral supernatant to transduce a patient's peripheral blood cells. And then those cells are expanded and infused into patients in the same way that we infuse TIL into patients. However, one of the things that we saw in this trial was toxicity. This is a patient who received T cells that express a very high avidity receptor, and we saw these severe rashes on the patient's arm as well as on this patient's trunk. Now, that did resolve. Eventually, the skin came back and had a completely normal appearance. However, this is obviously a very severe reaction. Patients have also had eye and ear toxicity. Some patients have actually had to take steroid eye drops for years after we've given the cells. So the cells remain in the patient's body. They actually remain active and can attack tissues in the body that have pigment, such as the eyes and the ears and skin. So we changed our strategy now and targeted other types of antigens. There are cancer germline antigens, which are, I think, more attractive targets because these are really limited in expression to germ cells. So they're expressed in the testes. They're not expressed in any other normal adult tissues. But they're also expressed in a fairly high proportion of multiple cancer types. So some of the antigens that we've targeted are NY-ESO-1. This is a relatively small family that includes 2 genes, as well as MAGE. This is a larger family of genes. And as I said, they're expressed in a wide variety of tumors. Here you see the percentage of tumors that express these antigens in a variety of tumor types. So bladder, non-small cell lung cancer, melanoma, ovarian, hepatocellular, myeloma, or squamous cell carcinoma. So many of these tumors express fairly high levels of these antigens. So one of the clinical trials that I've been involved with over the past several years is a trial targeting the NY-ESO-1 antigen. So this is a member of this highly restricted family of antigens that's expressed in melanoma as well as a number of other tissue types. As you can see here, we have an objective response rate of over 50%, and we have 3 complete responders out of the 17 patients that we've treated. Actually 4, but this patient subsequently recurred with another tumor. So a fairly high response rate and some very good long-term responders. And these are responders that have had disease in lymph nodes as well as visceral tissues. Here's an example of a large axillary lymph node. You can see this very large lymph node that has now regressed after 9 months, and this patient is now disease-free after more than 4 years. A patient here had a large liver metastases that was basically gone or nearly gone at 16 months, and this also is a long-term responder. And we've also treated other cancer types, and so this has helped us to move outside of melanoma, which has been the primary cancer that we've treated. We've treated a number of patients with synovial cell sarcoma, and here you can see we don't have any complete responders, but we have one patient who is now has some residual disease, but is doing very well more than 2 years after treatment. Generally, patients with this disease have lung as well as lesions in the bone, and these can be very large, as shown in the next slide. This is a very large bony lesion. This is the pelvis of this patient. This patient had a lot of difficulty walking because the tumor had invaded her pelvis. It was very painful and very difficult to walk. And as you can see, after 18 months, this tumor has shrunk down. You can even see regrowth of the bone here in parts of the pelvis. So the body actually can regenerate some damaged tissue, and this patient is now walking and doesn't have pain, doesn't have the problems that she had before the treatment, and hopefully that'll continue to shrink, and we'll see a complete response eventually. Now we can also target cell surface antigens using antibodies, and the way that we do that, these are called chimeric antigen receptors, is to identify an antibody that recognizes a cell surface molecule and actually engineer the molecule. So the antibody variable region domains, the portion of the antibody that recognizes the antigen, is engineered into a construct that contains signaling domains for the T cell. And so now that cell can be redirected against a cell surface molecule expressed on the tumor cell. And this has been fairly successful with one cell surface marker, CD19, that's expressed in B-cell malignancies. We have several actually long-term responders now in an ongoing clinical trial. But we're also targeting EGF receptor variant 3, which is a mutated gene product in glioblastoma, as well as mesothelin. So I just want to finish up with a couple of slides talking about our efforts to target mutated antigens. So we think that the most specific reagent that we can develop is a reagent that will be directed against a mutated antigen, because that will be specifically expressed in that patient's tumor. That will be a foreign antigen, basically. And so the way we're going about this in a project that we started with Yardena Samuels, who'll be talking next, is to sequence the patient's tumor and normal DNA and identify the somatic mutations that are present in that tumor, and then to predict which peptides or which genes we might be able to use to identify either naturally occurring cells or to actually raise cells from patients or from mice that recognize those antigens. And then those T cells can then be used, or receptors from those T cells can then be used to develop personalized treatments for patients. Now, one of the things that we, as well as others, have seen is that melanomas, which generally have a much higher mutation rate than other tumors, themselves can have very highly variable mutation frequencies. So these are the number of non-synonymous coding region changes that were identified in the tumor, and they range here from almost 2,000 to about 30. So there's a lot of heterogeneity in melanomas. These are all melanomas, and that's, I think, going to be a big challenge to understand what are the key genes that play a role in developing tumorigenic phenotype. And Yardena will certainly tell us something about that in a few minutes. But we've also used this approach to screen naturally occurring TIL for their ability to recognize these mutated epitopes. And using this approach now, we've identified 11 different mutated epitopes that are recognized by patients' TIL. And we've identified them from 7 out of the 8 patients where we've carried out this screening assay. So we don't know if this is associated in any way with the response. We think these are important for the response, but at this point, we've analyzed too few patients. You'll see here a couple of these patients were non-responders. One of them, however, did respond to at least one mutated epitope. But it's still very preliminary, and we're continuing to screen these patients to see if we can identify even more mutations, because as you can see, some of those tumors have hundreds or even thousands of mutations. So it's a challenge for us to identify all of the potential mutated products that could be recognized by that patient's cells. But that's basically where we are. So just to sum up, I think we can say that adoptive therapies with TIL, as well as genetically engineered cells, are effective at mediating responses. Targeting antigens with restricted expression should certainly facilitate this. But really, mutated antigens may be better targets for therapy than self-antigens, as we think they probably trigger more effective responses. And development of methods to target mutations involved with maintaining the tumorigenic phenotype or driver mutations may lead to even more effective therapies. And that is what Yardena will be talking about next.
Thank you very much, folks. Do you have some questions you'd like to ask right now?
They don't have to be cell surface molecules, because T cells recognize peptides that are actually produced by degradation of proteins. So antibody therapies require the use of a cell surface molecule, but T cells can recognize processed peptides that are endogenously cleaved within the cell. So it doesn't require a cell surface molecule. Yes?
What do you think of
Just a sec.
what do you think minor individual coincidences, whatever it is, it's not 100%?
Not 100%. Well, there's other genetic factors as well as sun exposure that are going to determine that. And I think people with that disease probably tend to stay out of the sun. I mean, they protect themselves from the damaging effects of UV radiation. So I would presume that's part of it. But it's a more complicated thing than just that.
Paul, do you have any comments about this Australian controversy of whether sunscreen actually does anything?
I don't really know. I think the best advice is probably just to stay out of the sun, because I don't think anything can be completely protected.
Thank you very much for inviting me today to give this talk. And indeed, we are focusing on the genetics, trying to search for new therapeutic targets. And so I'm not going to go over the melanoma as a disease because Paul went over that. I'll be emphasizing more genetics. And really, the paradigm for the genetic development in cancer comes from colorectal cancer. So you can see here where you have colorectal cancer developing stage-wise histologically. You can see how it is linked to various molecular changes at each one of these stages. And so really this has become a paradigm for solid cancers where each stage has additional molecular changes. And so what we know about it, we've known a lot, we've learned a lot, but we're trying to identify much more. And so that's really our aim. And we have good proof that it's worth looking at these genetic changes because you can see here some examples of alterations that have been identified. And here's BRAF, Paul was talking about it, which is highly mutated in various cancer types, especially melanoma at 50% of cases, which was identified by the Sanger team. Now, of course, we know that there's vemurafenib, which is FDA approved and is specific for the BRAF mutation. PIK3CA, which encodes PI3 kinase, also found to be highly mutated, is second most mutated oncogene in human cancer. Again, this is a target that many are trying to inhibit. So this really proves that it's worth looking for these somatic mutations in order to identify new targets for therapy. So in order to do this kind of analysis, looking for mutations, we first need to establish a tumor bank. And so I'll give you here a few slides explaining what I mean by a tumor bank. So here we see, this is for breast cancer, but of course it can be applied to various cancer types. So in this case, you have a primary breast tumor, which in some cases you can turn into a cell line, or you can put it as a xenograft within a mouse. And the advantage in doing this is that you are expanding the number of cells from which you can make genomic DNA. You're also really cleaning it up from the normal cells that are surrounding the like the stroma and fibroblasts, so that you get a population of cells which is much more homogeneous. And then you can actually extract the genomic DNA and analyze it for sequencing. This is just showing that you can take early lesions and later lesions and use those in your tumor bank as well. Now, a tumor bank can be made from different sources of DNA. So you can see here that you can take a frozen tumor, otherwise known sometimes as an OCT block. You can use paraffin-embedded tissue. Most of the tissues are paraffin-embedded, especially as pathologists look at it. And then you can make a cell line. Not every cancer type you can turn into a cell line, but we're pretty lucky that in melanoma there's a very high number of tumors that can be turned into a cell lines, about 50 to 70% success rate. And like I said, the xenograft from a mouse. And the advantages and challenges in using each one of these sources. So, of course, if you're using the fresh tumor, it's going to be reliable data because it's right out of the patient and it hasn't been processed, for example, making it into a cell line or a xenograft. But of course, you're going to have limited amount of DNA from the fresh tumor. It's going to be heterogeneous because, like I said, you haven't expanded particular clones of cells. And it's going to be a labor-intensive extraction because if you want it to be clean, you may have to have macro dissection or micro dissection or even laser capture micro dissection and a pathologist involved. Again, paraffin-embedded tissue is great for your data, but again, you get limited amount of DNA. It's going to be heterogeneous, labor-intensive, and the DNA quality sometimes is not very good. It's hard to apply to various sequencing methods. The cell lines, of course, you can get a lot of DNA. It's going to be much more homogeneous. The extraction is very straightforward, and you can use it actually for functional studies. which is a very powerful follow-up when you identify the somatic mutations. But you do need to go back to the fresh tumor to validate that what you identified in the cell line is actually here in the fresh tumor as well, to make sure that you're not following up on any artifacts. Again, xenografts, similar kind of advantages to the cell line, but it's very expensive, and you may have mouse DNA contamination, so you have to be careful about that. Okay, so of course, if you're looking for somatic mutations, you're comparing the tumor to the normal cells in the body. Usually you do that using blood, but that's not always available. So you may take neighboring tissue, but the problem with neighboring tissue is that with the sensitivity of current sequencing technologies, you may be picking up contaminating tumor cells. So that's a challenge. Of course, in order to get a good tumor bank, you have to try and gather as much clinical information as possible from the patient. So the date of birth, date of death, date of diagnosis, the malignancy stage, location of the tumors, the metastatic tumor, and also the therapies that that patient underwent. So of course, if that patient underwent radiation or chemotherapy, those may have induced mutations in their own right, and so you don't know if those have a role to play in the disease or they're just bystanders. So when I established my lab here 6 years ago and I decided to start working on melanoma, we were very fortunate to be able to work with the Rosenberg group and Paul Robbins and really set up an excellent tumor bank. And then after that, we've expanded to work with additional groups in order to have samples derived from different clinical centers. And this is really a summary of the tumor bank where we have metastatic tumor DNA. So these are late-stage metastases. We have the normal DNA, and that's usually blood. We have the original OCT blocks for all of these samples, so we can go back to them if we ever want to validate any of our data. For the Rosenberg group, we have matched cell lines, so we can derive from those RNA, match protein lysates, and perform functional studies. And for all of these, we have clinical information. And I'm emphasizing again that it's important when you're doing these kind of studies to look at additional groups, because it's good to validate whatever data you get in one clinical center in another clinical center, because there may be some biases because of patient recruitment, for example. So once we established our tumor bank, we performed several quality controls to make sure that the data we get is reliable. So we first make sure that the tumor and the normal match, meaning that the tumor and the blood came from the exact same patient using SNP analysis, because there are errors sometimes, and we don't want to follow up on polymorphism thinking that this is a somatic mutation. We also make sure that the cells that we're looking at are mainly melanoma derived, so we look at antigens that are specific to melanoma and make sure that at least 75% of the cells are expressing them. This allows us to be able to find loss of heterozygosity. And then finally, we sequence well-known oncogenes and tumor suppressor genes and make sure that the frequency that we identify them in is similar to what's known in the literature. And once we did all of this, we were happy with the tumor bank. We could go ahead and start sequencing novel genes. So just to recapitulate what a somatic mutation is, so you have your patient, the tumor and the normal tissue from which you could make a cell line, extract the DNA, sequence the gene of interest, and you do the same for your normal tissue and you compare these sequences. Only these somatic mutations are the ones that are interesting to us, and these are the ones we follow up on. To begin with, we've only looked at exonic sequences, so the ones that alter the protein amino acid. Now we're starting to look at the non-exonic somatic mutations as well. So we started off by sequencing single genes, by Sanger sequencing. We then moved on into sequencing families of genes, and now we're doing whole exome, whole genome analysis, which is much less biased. So this is, I showed you at the beginning, the progression of colorectal cancer. Also in melanoma, you have such a progression where you have the melanocyte within the epidermis, and it will develop into nevus. And it may develop to these additional stages where you have a dysplastic nevus, a radial growth phase, still remaining in the epidermis. At that point, it may invade the dermis and then disseminate as metastatic melanoma. And also here I'm showing you some molecular changes that are well known. Of course, by now we know many more of these changes, but our aim is to identify more of these, and like I said, we focus on this stage, the metastatic stage. So like I said, we're looking for these mutations. We don't stop at the genetics. We really believe that it's important to understand what these mutations do to the function of the protein. And we do try to be relevant for the clinic. So if we have a lot of choices in terms of somatic mutations, we'll probably choose the ones that we could apply back into the clinic. So I will give you two examples of studies that we've performed. One is looking at the kinases, the tyrosine kinases, which we chose to look at because, and so here you're seeing the human kinome, and the ones surrounded are the tyrosine kinases. We decided to look at this family because they're known to be highly mutated in different cancer types, and they're known to be mainly oncogenes, and they are amenable to pharmacological inhibition. And like I said, we want to be clinically relevant. And so this is just the screen that we did. We always do our screens in a two-stage manner. We start off with a discovery phase, and then we move on to a validation phase. The discovery phase is done with a smaller number of samples. So in this case, we used 29. We looked at 86 genes, the 86 tyrosine kinase genes, and we only sequenced one area, which was the kinase domain, because if there's a mutation, one would expect it to be there. And any gene that had one somatic mutation or more moved on to the next stage, the validation stage. There were 19 such genes, and we sequenced them in additional samples, 18 total. And what we found was one gene that was highly mutated, and that was ERB4. And this is just a scheme of the protein and the various mutations that we identified. And so this is an example of a Sanger sequencing high-throughput method that we did in 2009. And so we then moved on to understand what these mutations do. It was so highly mutated. So what is ERB4? We know that it's part of the ERB family, which consists of 4 members: the EGF receptor, HER2, which both are known to be good targets in the clinic, ERB3, and then ERB4. So these either homodimerize or heterodimerize on the cell membrane, and upon ligand binding, they will transphosphorylate, forming phosphorylation sites, docking sites, for various signaling molecules. So Todd Prickett in my lab cloned 7 of these mutations based on bioinformatic analyses that we could go into later in my talk if you're interested. But then we did a basic kinase assay to see what do these mutations do to the kinase activity of ERB4. And we compared it to wild type and kinase dead. And you can see here that there's an increased signal for all the 7 mutants, meaning that they are more active compared to the wild type, even though they're similarly expressed. So this was pretty striking to us. And so we did multiple studies to look at these mutations. I'm touching upon very few of these, but so this one, for example, is to show that we could use the melanoma cell lines in which the mutations were found and modulate those cell lines. In this case, we used shRNA to knock down the endogenous ERB4. So you see that ERB4 is lost. And then we made these stable cells, and we checked how does the knockdown affect the growth of the cells. So we did this in cells that were either wild type for ERB4 or mutant for ERB4. And when we did this in cells that were wild type for ERB4, we didn't see any effect on their growth. You see, it's similar to the vector control. But when we did this in cells that were mutant for ERB4, they did not grow so well. So this suggested to us that this could be an interesting target because if you're knocking it down specifically in mutated cells, they stop growing. And this is called oncogene addiction. So the next step was to see if we could use a small molecule inhibitor that's currently being used in the clinic. And so we used lapatinib, which is FDA approved, and it's being used for breast cancer patients. And we exposed multiple cell lines, these are only a few of them, that were either wild type or mutant for ERB4. And you can see here the various concentrations of lapatinib. And when we plotted the survival data, we could see that the ones that had a mutant ERB4 were significantly more sensitive to the exposure to lapatinib compared to the wild type cells. So based on this study, we hypothesized that about 20% of melanoma patients are going to harbor their ERB4 mutations, and they may be sensitive to lapatinib. And at that point, Udo Rudloff came to my lab. He was a recruit at the surgery branch, and he came to my lab for a few months, reproduced all the data, and wrote a clinical protocol that was performed here at the NCI, at the surgery branch, in collaboration with Memorial Sloan Kettering. And the sequencing of patients was done by Paul Meltzer at the NCI, and this is really the protocol. And so patients were being screened for ERB4 mutations, and anyone who consented and had the mutation got the lapatinib. What we do have from this study is that if these mutations were the ones that I showed you to begin with, these are the ones that were identified in the clinical trial. And what we see here is that the frequency is similar to what we identified to begin with. And these are additional whole exome genome samples that we have sequenced since our first paper. And you see that we're still finding these mutations. And this particular study was by Nick Hayward in H Genetics, and you see additional mutations in ERB4. And finally, this is another paper publishing ERB4 mutations. What we can see now is not only that the frequency is high, but we're starting to see recurrent mutations in particular locations. Some of these were functionally evaluated, the ones with the stars, and so we know they're activating. What this means, we still don't know, but it does seem to be highly mutated, and it does seem to be playing a role in a melanoma progression. Okay, so then we moved on with technology, and we started using second-generation sequencing approaches. And so I'll give you one example of those studies. And this is a whole exome study where we captured, and we were the first to publish melanoma whole exomes. So this was in 2011, so at that point it was still very expensive, and so we only did 14 samples, but it was a very good proof of concept. So we had 14 tumors and 14 matched normals that we captured using the Agilent SureSelect. At that point it was a 37 megabase, and we sequenced using the Illumina platform and then used ELAND to look at the data. Any of the validation, was done using Sanger sequencing. So we like using a different method to validate the data, an orthogonal method. This is just an example of how the capture works. You can see here the oligos that are capturing the BRAF, and particularly BRAF here, and you can see the sequencing depth for the various exons right here. So you see there's a variation. And so we do need to go to sufficient depth in order to look at the alterations in each one of these positions. And so you'll see that we go to over 100x coverage. So this is the performance for our whole exome. We get 12 gigabase of sequence for each sample. Like I said, our mean depth was 180x or greater. And so we have a very high coverage, very low false negative rate, and our sensitivity is 81%. So these are the steps that we took for this study. Like I said, we looked at 14 untreated samples. We got a number of potential somatic mutations. That this was prior to filtering. So we assemble the data, and then we filter out the somatic mutations. And then for this particular study, we developed a new algorithm called MPG to coverage ratio. The MPG score was already published prior to this study, which is the most probable genotype. In any case, we took into account the coverage of each one of these samples, and we found that when we applied this new algorithm, we could reduce the number of somatic mutations. So we removed 18%. And this is the way the mutations divided: missense, nonsense, insertions, deletions. We always look at synonymous mutations as well, not only the ones that affect the coding, but also the synonymous, because we want to look at the ratio between the non-synonymous to synonymous mutations, because if it's not significantly above what's expected by chance, these are probably neutral effects. They're not really affecting the tumor. And so you see the ratio is 2 to 1, which is not significantly higher to what's expected by chance. So most of these alterations are passengers, meaning they don't have a role to play in melanoma. And that's really the challenge. And as Paul was talking, some of these melanomas have an extremely large number of mutations. And so it's really hard to find out which ones really play a role in the disease and which are just neutral. They're just because they were exposed to UV and they just have these alterations. And so that's what we're trying to work on now, to apply various methods to see what are the drivers in the disease. And so the statistics, we look for hotspots, meaning the same exact mutation again and again and again in the same position in different samples, such as seen in BRAF. We also look for the frequency, so if it's highly mutated, it suggests it's being selected for. I told you about the non-synonymous to synonymous, and we look at the background mutation rates. So when we looked for hotspot mutations, we found 9 such genes. And that was surprising because we knew of BRAF, but we didn't expect any additional recurrent mutations. So again, we validated. We look at an additional sample set to see whether this reproduces and scales up. And it did for at least for one of these positions, which is called TRAP. This is a gene called TRAP, and you see the same exact alteration occurred in 6 different patients. And this was also seen in other studies, as you see here. And so we're now working on this particular protein. We know that it does affect the cell survival in the cells in which it's mutated. So this is just an example of how, through genomic data, you can find these novel recurrent mutations in genes that we had no idea that had a role to play in the disease. And this is the other side of the coin, looking for highly mutated genes. And so we found 16 highly mutated genes, and again, we validated them in more samples. And it's a condensed slide with a lot of information, but what the take-home message is that when we do this statistical test, we do find BRAF to be on the top of the list, which is to be expected, with the lowest p-value, the most highly mutated, but you find other genes in this list as well. And then when you validate it in more samples, the frequency remains similar, so it's concordant. It's suggesting that we're doing something right here. And so this GRIN2A was the second most highly mutated gene, and we found it fascinating because it was never linked to melanoma before. And so if you look at GRIN2A, this is the protein. These are the mutations that we identified, and the boxed ones are nonsense mutations. These mutations truncate the protein. And so this occurs in about 15% of all the mutations, which is really a definition of a tumor suppressor gene, if you get these nonsense mutations. So something is going on here. This is probably a tumor suppressor gene. This is an ionotropic glutamate receptor, and I'll touch upon this particular pathway later in my talk. Just to say that some of these mutations have already confirmed in other studies, and additional ones since as well. So in addition to looking at genes, single genes, since we are looking at the whole genome, we can start looking at pathways as well. And there are many different ways to do that. And the first person who did this was Bert Vogelstein and his group, where they looked at all the exomes in pancreatic cancer, and they found that there were 12 core pathways. So even though 2 different patients had completely different mutated genes, you could start noticing that these genes actually fall into the same pathways. And so when you start deciphering that, then you can't develop therapies to each one of these mutations, but you can't try and develop therapies to different pathways. So this is the optimistic side of looking at all of these mutations. And so when we had our exomes, we did a similar analysis, and we found one pathway to be significantly mutated, and the p-value is here, and that's the glutamate signaling pathway. And when I say glutamate signaling pathway, I told you about GRIN2A, which is an ionotropic glutamate receptor. It binds glutamate, and you can see these various proteins that are highlighted. These are the ones that we found mutated. So I told you GRIN2A, ERB4 I told you about, and there's actually a paper in Nature Medicine showing that GRIN2A and ERB4 interact, and ERB4 phosphorylates GRIN2A, and we have recapitulated that in melanoma cells. And then we have other genes here, and we don't have the time to go into them, but this is a pathway that is fascinating to us. So we're going to continue sequencing more melanoma patients, and that's because if you look at the mutation frequency in melanoma compared to other solid cancers, you see it's extremely high. And I'd like to emphasize this is an average of many melanoma. So of course you see a variation between them, but it's still extremely high. It's about 16 to 20 mutations per megabase. So we think that we need to analyze many more of these to find out which are the drivers. And so that's what we've been doing. We've been also merging a lot of data from other sources, because this has been a very fruitful time in the melanoma field. You see there have been many, many studies published in the last 2 years, genomes, exomes, and so we've been merging this together with our data and other unpublished data from other groups. And at this point, we have a very large number of melanoma cases that we have merged, and we can see that there are recurrent mutations. They keep extending, and you see that positive controls come up on the top: BRAF, NRAS, RAC1. These are all known hotspots, but we have more to look at. And so that is really our aim. So we're looking at these, and we're looking at them at a total of 600 samples in order to gain frequency. And we have some interesting targets already that we're validating at this point. So in addition to all of these groups that are sequencing exomes and genomes in melanoma, of course, there's TCGA, the Tumor Cancer Genome Atlas, that was launched in 2006 as a pilot. It's ongoing, and it actually included melanoma. And I'm on that work group, and we actually now have over 200 melanoma exomes through that initiative. So we're going to have a very large amount of data to look at very, very soon. This is just to show what TCGA does and the fact that indeed they are looking at DNA sequence as well as many, many other aspects of these different cancers. So I'll cut short because it is becoming late, but our challenges now are still going to be looking at the drivers and the passengers. We want to keep analyzing, but it's a challenge of how to interpret the data. We do aim to do functional analysis, but how do we do it in a high throughput fashion, and how do we apply this to the clinic? And so if now we're mainly looking at the disease in the clinical and pathological aspect, and this is really going back to the way we started the whole afternoon, we now hope to be applying more mutation analysis in order to find the various melanoma subclasses of this disease, in order to further develop more targeted therapies that may be more specific to the ones that we see here in the slide. And of course, the TIL therapy that Paul was talking about is extremely exciting, and hopefully will lead us to better results in the future. And these are many, many people that have contributed to the work. Thank you.
Thank you very much. We have some time for questions for either speaker or general. I would ask you to speak loudly because there are many people who are online, and they send me emails wanting to know what the questions were sometimes. Are there any questions?
I wonder if there was something unusual that you saw from your exome sequencing on amelanotic melanomas as compared to the more common pigmented type.
We haven't categorized them based on the melanin content, so I can't answer that question. I'm sorry.
So is it well, in the absence of all the data, it's hard to, you can't draw conclusions, but you can certainly get impressions that influence the way you think about it. So this question may be a little bit strange, but when you look at the histologic pattern of changing, let's say, of melanoma from an initial mole eventually to an invasive thing, I mean, classically, this is what's described as specific stages. Is it your kind of feeling that in any given patient there are likely to be common drivers for those stages, or is it more complex in that there are many drivers interacting with many different kinds of machinery?
It's a great question. So because I don't think we have enough data to completely answer that question. Now that we're going to merge, hopefully at least 400 exomes and genomes that include clinical information, But again, these are all metastatic, so we don't have the primary or earlier stages. Hopefully we'll be able to divide those into subclasses based on mutations, right? And link that to at least some of the clinical and pathological information that we have. We're hoping in the future, as technology develops and you can start doing sequencing with less DNA and analyze it even though it's heterogeneous, we'll be able to pick the most interesting targets and sequence those in earlier lesions, okay, which are difficult to come by, but you know, we have been able to get some of those, and then be able to see which ones of all this compiled list of mutations occur earlier and which ones occur later. Do they have similar drivers? Well, if you look at BRAF, for example, you find it in nevi already, and it's pretty frequent in nevi. So that's an early stage, and that's a common denominator. So there you have an example where you have a driver that does occur in 50% of the melanoma population. But whether there'll be more like those, it's still a question. Paul, do you have anything to add to that?
How do you control, I'm sorry, Bob.
Go ahead.
How do you control for the fact that most patients, if they're found to have a melanoma, are treated in one way or another? I mean, on the outside, they may get chemotherapy, I'm not sure. But how do you control for the fact that when you have the tissue and you do all the genomic analysis, how can you discriminate between what is the response of the tumor to the therapy that was given, both beneficial and maybe resistance, and the fact that all the patients, you know, are all different in that regard? How do you control for something like that?
Well, at the moment, there are not that many targets and not many inhibitors. So there's only like a one-to-one correlation, I think. So if you take BRAF as an example and you give emurafenib, then you're just following up on that particular response. I think, am I understanding your question correctly? Yeah, and like Paul was saying, they are doing now some they are merging different drugs together, such as vemurafenib and MEK inhibitors, and that is increasing survival by 3 months. But that is really our dream, to be able to have off-the-shelf inhibitors that are FDA approved, have the sequencing information, and based on that be able to mix and match and see whether the tumor is more responsive.
Yeah, we started originally talking about radiation, sun radiation, as one of the drivers, and you talked about some of the mutations perhaps not being drivers within melanoma. Do you get a sense that all the drivers that lead to melanoma are radiative in origin, or some systemic toxins that have caused the mutations? Or is there some combinations of them?
So there is a C to T signature, which is a UV signature, and even more so a CC to TT UV signature, which, so if you start looking at these mutations and just look at those signatures, you'll find that there's significantly more C to T alterations. Not all those are drivers. So obviously you have all of those bystanders just because of the UV. On the other hand, BRAF, which is the main driver in melanoma, does not have a C to T mutation. So so based on that, people started to say, okay, well, if you look at a mutation, it's a C to T, it's probably a passenger. It's not going to be a driver because of BRAF. But recently there has been a Cell paper by Linda Chin and colleagues showing that actually drivers do also have this in this C to T signature, so you cannot exclude them as playing a role in the disease. Now, in addition to UV, you have other toxins, as you were saying, that could be inducing other kinds of toxicities. And I think that David Fisher recently had a Nature paper dealing with that. Does that answer your question?
It goes there. I was sort of just trying to, you know, if somebody grew up in a cave, never got exposed to sunlight, doesn't have a UV driver at all, is there still going to be melanomas? And if so, what's the drivers behind them?
That's a good question. So that's what David Fisher just published, that even in the absence of UV, and we were thinking, okay, no exposure to UV, then we're going to be protected, or if you use sunscreen. Unfortunately, that is not necessarily the case.
Well, I can comment. I mean, some of the melanoma samples that we've looked at don't have predominantly C to T predominance. So, like on that slide that I showed, where the variable frequency of these mutations, a couple of the tumors that have fairly low numbers of mutations don't have a predominance of C to T mutation. But that's where I think the clinical sort of data is also important. That tumor, I believe, arose on a foot or heel. So it may be even the location of the tumor plays a role, right? If it's a sun-exposed part of the body, then it's going to be more exposed to ultraviolet radiation. So that introduces more complexities into the whole situation. So the question of the same drivers driving that tumor that was present from a patient's foot primary, are those same drivers driving the tumor that developed on a patient's scalp? But that's why studying these large panels of patients and correlating that with clinical characteristics, I think will be fascinating, because only then are we going to be able to really make some sense of it. But that's why that study is so important.
So is there a racial difference in the epidemiologic occurrence of melanoma? Does having melanotic skin protect in any way, or accelerate, or have no effect?
Frequency is much lower. Go ahead, Paul.
Well, yeah, the frequency is much lower in African Americans, and of course it's affected by the color of the skin, and the color of the skin, of course, is affected by mutations also in the pigment genes that give rise to the melanoma. So that as the populations, presumably evolution occurred by migration of populations from Africa or parts of the earth that were warm, and as those populations migrated north, there was less of a selective pressure to maintain darker pigmented skin because there was less UV radiation, so it had less of a disadvantage. So those genes then could mutate, but of course they then created susceptibilities, presumably, to cancer if patients were exposed to ultraviolet radiation. So there was perhaps somewhat less selective pressure, but nevertheless, those mutations still presumably predisposed to mutations. But again, you need to really sort it out, you need large studies, right? So these large studies, like Yardena was talking about, will help to sort it out, presumably, because you'll have all the patient characteristics, you have the clinical histories, and then you can sort out the types of mutations that occur in various populations, patients that have fair skin, red hair, patients that have, you can perhaps start to parse that out. But you need a big study to do that. You can't just study a small number of patients, you won't have enough data.
Okay, are there any other questions? Well, if not, I want to thank you on behalf of all of us for a really very exciting glimpse into where things are going. Thank you. Continue this work.